I'm Gretchen Kaiser. I'm the Executive Director of the Research Development Office. I know a lot of you, and I want to welcome you this afternoon to our Marcus Mixer 2. I just want to say that we're excited to be able to continue this program, and really excited to hear about some of the progress today. And I'd like those of you who've had projects funded and are here to sort of hear each other's research, to also keep your minds open to some of the other talks we have today that have to do with some of the cross-cutting disciplines that could make for some really interesting contributions to some of our basic and our translational research projects, things like bioethics and health disparities, etc. So I welcome you all. Thank you for being here. And without further ado, I will introduce Keith Yamamoto, who I think amongst this crowd doesn't need any real introduction, and he'll tell you more about the program this year.
Thanks, Gretchen, and thanks everybody for coming. This is a really exciting program, in no small part because of the vision of the donors, George and Judy Marcus. You all know that precision medicine is sort of the central operating principle for the whole campus, in which the goal of that endeavor, despite the funny name, is not for everyone to do medicine, but for everyone to develop in their own areas of interest an understanding of a biological or pathological process, and then to weld all those pieces of information together in a computational knowledge network that will aid all of us and provide information about mechanisms of disease for patients, ideas for new hypotheses for basic scientists, and really aid the whole endeavor across the board. Well, George and Judy Marcus understood that, and so what they wanted to do with this program in precision medicine innovation was to seek out projects that would really have a basic science question or issue or problem at its base, but could extend all the way to, or at least toward, the capacity to improve patient outcomes. In many cases, or most cases, from our experience, that means that a basic scientist will collaborate with a clinician scientist or a clinician to develop the project and put together the proposal. And we were excited to see and not surprised, to see from the ECSF community fantastic proposals of that sort that really filled that challenge from the Marcuses to begin with this foundation of a basic science problem and then to move forward from there. We'll hear some examples of that in the lightning talks today from past award winners and from some others in the community that are kind of filling, as Gretchen said, kind of filling some of the gaps that we really need to be attentive to in really carrying out this precision medicine mission. We find all of that very exciting, and the opportunity to continue the program through the generosity of the Marcuses putting in another $2 million into the program really speaks to their confidence in all of you, and the capacity for the UCSF community to continue to really build on that principle. Just to give you an idea, for those of you who aren't already familiar with the program, the funding program is divided into two sectors. There is a Seeding Bold Ideas program, just a new idea, doesn't have to have preliminary data behind it, something that is exciting and has great potential, just a one-year grant for $75,000 in general for two or more investigators. It doesn't have to be that way, but that's usually how these things succeed. And then the second grant program is called transformative integrative research. It is in the range of $250,000 to $400,000, so it's for established programs that are underway, where you can really show that opening a pathway with this funding would really make that program move forward in a transformative way. Both programs are 1 year, but they're renewable, and the Seeding Bold Ideas grants that are successful are fully eligible to apply for the large funds in the transformative program, 250 to 400 in the second year. So it's just an open competition actually both years. The transformative people will be most likely applying for a renewal, and then the seed program people will be coming in to take their money away. It does happen. I can tell you. And then just to give you an idea of the statistics, so you can see where your chances of getting funded are. In 2016, when we started this, we received 25 Seeding Bold Ideas initiative applications and funded 8, and 20 transformative integrative research and funded 4. So a lot better than the NIH. In 2017, 21 Seeding Bold Ideas, 7 funded. 21 transformative integrative research, 3 funded. So not bad statistics in the day and age that we're all operating in. Just to give you an idea. So we're excited. I want to thank you all for coming. Hope that this event stirs interest in you and making contact and networking with others. You probably are walked in with ideas pretty firmly in mind, but if not, there may be people in the room that you will be able to touch base with to try to develop something that you've had on your mind, but you don't have the other half, either the basic or the clinical translational piece in mind yet. There may be someone in the room that could fill that bill. So hopefully the mixer part of it can work just as effectively. So with that, let me stop. Thank you all for coming, and turn it back over to get the lightning talk started.
Thank you, and we'll have our first speaker, Dr. Chloe Atreya. So it's this one. Okay. Perfect.
Hello. So my name is Chloe Atreya, and our project is entitled Kinome-Guided Targeted Therapy, Targeting Pro-Inflammatory Cascades in Patient-Derived BRAF V600E Colorectal Cancer Xenografts. And our team came together to try to improve outcomes for patients with BRAF-mutated colorectal cancer who have a particularly poor prognosis. I'm a medical oncologist with a PhD in pharmacology, and I've led several of the clinical trials for this population, including the study on the bottom right. That was one of our more successful clinical trials with a response rate of 21% and a median progression-free survival of 4.2 months, which illustrates that there's still a lot of room for improvement with this population. And the patients on the clinical trials had pretreatment biopsies from which we were able to establish patient-derived xenograft models, which were used in this project. And I have partnered with Jean-Philippe Coppet, who is a molecular biologist who developed PhosphoAtlas and a high-throughput kinase activity mapping platform, which really enables both detailed as well as extensive characterization of what happens to signaling after treatment. And we're also working with Kayvon Shokat, a chemical biologist who is an expert in kinase inhibitors and signaling, and Byron Hahn, who manages the preclinical therapeutics core and is an expert in everything related to PDX models. So our project came out of some preliminary data from Jean-Philippe, where when colorectal cancer cell lines were treated with a BRAF inhibitor, a MEK inhibitor, and an EGFR inhibitor, he was showing parallel mechanisms of resistance that were driving the resistance to therapy, and these were specifically related to pro-inflammatory cascades with COX-2 and SRC-relayed inflammation. And so our Marcus project is designed to test the hypothesis that this is an important cooperative dependency for these tumors in patient representative PDX models. And so we did testing in several different treatment groups using the PDX with up to 4 therapies. And if 4 therapies were used, one of the therapies had to be celecoxib because it's particularly well tolerated. These are some of our most interesting results. So the 3 graphs show 3 separate PDX models from different patients. And the dots represent 8 different mice that were treated with a particular therapy or combination. And on the left are the vehicle-treated mice, the controls, that those tumors grew very large in a 21-day period. The next 2 lanes are the clinical combinations, dabrafenib, BRAF inhibitor, with the MEK inhibitor trametinib, with or without panitumumab, the EGFR-targeted antibody. And then our most interesting data was with the addition of celecoxib to dabrafenib, trametinib, panitumumab, and those 3, so that quad. That's where we consistently saw the best tumor growth inhibition, and in some cases it was statistically significantly improved compared to the other treatment arms. So we think that this is a promising result that is worth further investigation. And to do this, we have collected all of the tumors from the mice that we treated or that received the vehicle, and we'd like to go on to characterize these tumors, including using HTCAM, and to computationally integrate the signatures to figure out more about what's happening to the pathways in the PDX tumors, and then to start optimizing our combinations until we ultimately have a strategy that we think will provide a meaningful benefit that we can bring back to patients. So at the bottom is our proposed pipeline, and we're awaiting our R01 score. So. Thank you.
Thank you. And now please welcome Dr. Stephanie Christensen.
All right, so thank you for inviting me to talk. I am one of the pulmonologists, and this is work that has been through a collaboration with our infectious disease colleagues to look at improving diagnostics for acute exacerbations of COPD. Okay. So just to give you a little idea of the scope of the problem, so COPD is the 3rd leading cause of death worldwide, and acute exacerbations contribute greatly to that increased mortality as well as to morbidity. And despite the fact that it's actually a really huge problem, we have very antiquated ways of trying to diagnose and treat these patients. So the definition itself is just really very imprecise. So acute flare-up of patients with of their underlying inflammation. So these patients already have inflammation at baseline, so they've got diseased airways and microbial dysbiosis. And then we add to that a trigger, and sometimes that's a viral or bacterial infection, but sometimes it can be environmental, so say smoke from the fires recently. And then once they come in to visit us in our office or in the hospital, we pretty much always give them either steroids or antibiotics or both. It's not really evidence-based, and one can kind of guess that might actually cause harm in some people. Shocking. So our solution to this problem has used metagenomic deep sequencing of sputum samples. So, and this is kind of leveraging the work of Dr. Langelier, who has been doing something very similar with pneumonia. And basically, by deep sequencing these samples, we can get both the host transcriptome as well as possibly pathogenic microbes in the same samples. And we can use that data to both look at the host response, both at baseline and during an infection, as well as potential pathogenic triggers. So we were lucky enough to get samples from some of our collaborators in the Netherlands who just had a big old bank of sputum samples saved up from a study of exacerbations in which they had baseline samples, samples at the time of an exacerbation, and then after they were randomized to steroids or no steroids along with antibiotics. And they got sequential sputum samples over time. And so we can use that data to potentially, using our deep sequencing, to potentially develop biomarkers of both diagnosis and treatment response. And so I just wanted to show you, so we're still finishing up the sequencing. It's a very large data set, but this is just one of our patients or participants here in which, let's see, okay, great. So this participant, over time, we can see that this is at the time of baseline, and over time they develop parainfluenza virus. So this is at the time of exacerbation, and then this is 1 day later, and then it comes down over 2 weeks. So we can see that we do see a virus. You can also see down here we did some time series clustering of these patients. So not only are we looking at differential expression over the whole patient population, but we're going to look in a particular patient at which subsets of genes are particularly upregulated. And so this group of genes is actually more involved in oxidative stress and cell repair, whereas the genes that we saw in other patients who had bacterial infections, I don't see upregulated. They just kind of have a flat line over the patient. So that's just kind of a sample of what we've been doing. So this work leveraged, as I said, Dr. Langelier's work with pathogen detection in acute pneumonias, as well as Dr. Crawford's work in innovative sequencing methodology, and our work looking at genomic signatures to identify heterogeneity in COPD. And so it's been a really great collaboration. So our next step, so as I said, we're still doing some sequencing, and the idea is eventually to take this into another larger study that we already have underway with exacerbation, sputum samples being looked at for exacerbations in a multi-center study. And then we've also started with Dr. Lawrence Wang at the General, looking at the relationship between acute infections and COPD in relation to HIV, where both of those diseases are enhanced. Okay. Should I get it? Should I do that in 5 minutes? Yes. Thank you.
Thank you. And now we'll have Dr. Zimu Deng.
Hello everyone, my name is Zimu. I'm a postdoc working with Anthony Sham in pulmonary. Today I'm going to talk about a project in the Sham lab about a novel Mendelian syndrome called COPA syndrome. So this is an autoimmune disorder specifically affects lung and joints. Most of the patients develop interstitial lung disease and inflammatory arthritis. So most of the patients have autoantibodies such as ANA, ANCA, and they all respond to immunosuppression treatment. So here is an example of the lung biopsy from the patient. You can see that there are massive lymphocytes aggregation in the lung. So by whole exome sequencing, we were able to identify the gene variants that co-segregate with the disease phenotype. And this gene is COPA. It encodes a protein called alpha COP, which is a subunit of a protein complex called COP1 complex. So here. So this COP1 complex mediates the protein trafficking from Golgi apparatus to to endoplasmic reticulum. And this protein trafficking pathway is highly conserved from yeast to mammals. And the mutations we identified in the COPA patients are highly conserved in the COPA protein. So this suggests that by translating the disease to an animal model will be promising. So we decided to generate a mouse model carrying one of the mutations we found in COPA patient, and those mice spontaneously develop lung disease with a similar pattern to the human disease. So here is a simplified picture I'm trying to use to explain the cellular defect caused by those mutant COPAs. So in the homeostatic condition, there are constant protein trafficking between ER and Golgi. However, if the cell carry a mutant COPA, it will impair this retrograde transport. And this impairment will lead to ER, to the disturbed ER, and finally lead to a unique response called ER stress. So here on the right, you can see that in the mutant COPA cells, there are upregulation of the ER stress markers, BIP and CHOP. So the sustained ER stress will finally trigger inflammatory response. And then the cells start to secrete inflammatory cytokines such as IL-6, as you can see on the right, and L1 beta, etc. Again, like I said, this is a simplified schematic. There are still a lot unknown, detailed mechanisms unknown about this disease. So with the help from Marcus Foundation and collaboration with Dr. Waterfield, we were able to carry this study further by performing RNA sequencing. So our current goal is guiding a comprehensive understanding about the networking during the disease progression. And we got the data back right now, so Mike is very experienced in analyzing and interpreting the data, and we hope we can get some interesting hit. So besides all the mechanism work, we are also developing tools to target the ER stress pathway. So here on the left shown is a ER stress measurement using our assay. So it's based on luciferase, and the red bar you can see when we add ER stress inducer, there are about 3-fold change, and the green bar represent the effect of the ER stress inhibitor. And we have developed a high throughput screening use those methods, and we are testing the compound library together with Dr. Oak's lab. And the molecules coming out of this screen will be the potential treatment for COPA syndrome. So last but not least, I would like to thank the Marcus program for the support of this project, and especially this opportunity to present our work in front of everyone. I would like to thank my mentor, Tony Shum, and our collaborator, Mike Scott, for the discussion and the collaboration, and everybody in the Shum Lab for the hard work. And thank you for listening.
Thank you, and please welcome Dr. Sam Goldman.
Alrighty, good afternoon. I like this format. It plays to my short attention span, which is probably something a scientist shouldn't be bragging about. But so our project is called Toward a Redox-Directed Therapy for Parkinson's Disease, Integrating Environmental, Genomic, and Functional Data. And Raymond Swanson is the PI. So what's up? Charcot thought that Parkinson's disease, paralysis agitans, is not a family disease in 1877, and shortly thereafter Gowers came back with the observation that many patients do indeed have a strong family history. And now a century has passed, and we're pretty much in the same place we were. So a number of years ago, Carly Tanner and I undertook what we call the FAME study, which was a national study based in the Agricultural Health Study, which is a cohort of 50,000 professional pesticide applicators in Iowa and North Carolina. And we screened the entire population for Parkinson's disease, went out and examined them and matched controls, and we found 100 persons with PD and 370 controls, and we have very detailed exposure data on these folks, particularly with regard to their use of pesticides, and we collected DNA. So the results of the epidemiologic study was that we found a 2 to 3-fold increased risk of Parkinson's disease for individuals who had professionally used paraquat or rotenone. So this was a terrific observation. But what was really striking was that if we looked within individuals who either had this enzyme GST-T1, glutathione transferase T1, or didn't have it, and this is a very common deletion in about 20% of the population, so we had good power to look at this. So in individuals who had GST and weren't exposed to paraquat, that was the reference group. In those who were null for GST and weren't exposed, they had a very minimal increase in risk. For those who had GST and were exposed to paraquat, they also had a minimal increased risk. But if they were both deficient in GST and were exposed to paraquat, then they had this gene-environment interaction with a remarkably increased risk. So that's the EPI. And we presented that data at a neurology retreat here, and Ray Swanson was there, and he observed this, and he's a glutathione expert and has studied glutathione in a variety of models, and he has an animal model of Parkinson's disease, wherein glutathione deficiency results in a Parkinsonian syndrome. And we've long known that glutathione is essential somewhere in the Parkinson's pathway. And Ray had this great idea to bring in Steve Finkbeiner's research group and their expertise relating to molecular mechanisms of plasticity and neurodegeneration, and their use of induced pluripotent stem cells to look more deeply at variants in Parkinson's patients. So what we're doing is developing a personalized toxicology, in effect. So we're looking at specific toxins and specific genetic vulnerabilities. What we're doing that no one else has previously done, to my knowledge, is we're looking within subsets of exposed persons. So a subset of both cases and controls who were exposed to paraquat, a subset of cases and controls who were exposed to rotenone. And within those groups, we're looking for individualized susceptibility. So we've done, to date, we've done a million SNP array, and we've identified a range of genes that are exciting and interesting. We're using both an unbiased approach and a hypothesis-driven approach, so the glutathione enzymes involved in glutathione metabolism and pesticide metabolism. And the next step will be to conduct a targeted sequencing of genes that we suspect are playing a specific role in individuals exposed to these pesticides. And subsequently, and concurrently, we'll be modeling some of these variants using CRISPR and siRNA in existing iPSC-derived dopaminergic neuronal lines, and evaluating changes in parameters of redox metabolism in these cell groups. So thank you very much. Appreciate the opportunity to share.
Thank you, and now we'll have Dr. Barbara Koenig.
Thank you. That was a great talk, and I just wanted, as I was listening to it, my first thought was, what will you do if you identify workers who have a gene that might lead them to have a bad reaction from a pesticide, and that's the only kind of employment open to them at a particular point. So those are the kinds of dilemmas that I worry about as a bioethics scholar here. So I'm a sort of different beast than the rest of you. My name is Barbara Koenig, and I am a medical anthropologist by disciplinary background, but I work in the interdisciplinary space of bioethics, and I have the honor of serving as the director of UCSF Bioethics, having been given that appointment by the provost, and we're trying over 5 years to revivify our bioethics infrastructure at UCSF. And I have 2 appointments, just so you know how people like me exist, since I'm getting more basic scientists in the room. I'm primarily appointed in SBS, the Social and Behavioral Sciences in the School of Nursing, and secondarily in Anthropology, History, and Social Medicine. So that's who I am. And I have a number of points of intersection with precision medicine. I don't have a specific Marcus-funded project, but I'll give you sort of the kinds of things that I personally and with teams of other scholars have worked on over the last several years. And the first is I had a large R01 from NCI to look at the issue of family disclosure of genomic secondary findings. So when you do all these sequencing projects, you reveal a lot of information, and sometimes you get information that's relevant not just to the individual patient or participant, but to their family member. Like, what if you discover a really big actionable gene? So that's turned out to be a really problematic thing. Do you just call up the family members, et cetera? So that was one project. A second project was called Engage UC, and that was collaborative with Dan Doan and others here at UCSF. That was to look at consent issues and governance issues for the idea of having a biobank that would cover all the UC campuses. So that was a really interesting project. We did some actual studies of different ways of doing informed consent, but then of most interest we did what's called a deliberative community engagement, meaning that we got together small groups of members of the public in both Southern California and Northern California to get their views and input on what the governance of our biobank across UC should look like. So that's another project. Then the third bullet there is a CEER, meaning a Center of Excellence from NHGRI. So we also have had a Center of Excellence in Translational Science to sort of pair up with a lot of different projects and think through with the scientists what the issues might be. And then with Keith recently we've been thinking of a plan for renewing this CEER, and we hope to do that at some point, the Center of Excellence. And then the three current studies that I have funded, one is a study through something called the EnSite Network, which is a project looking at sequencing of newborns, which is a really complicated issue. Under what circumstances is it a good idea to do a full genome sequence of a newborn, either an ill newborn, for example, an undiagnosed infant in the NICU, or a healthy newborn? And if it's in the case of healthy newborns, how will that affect the standard newborn screening that we do on a public health basis in the state? So we've just written a big report about how sequencing intersects with newborn screening that will be published in a bioethics journal. Then next I have a collaboration here with Laura, and this is just an idea of the kinds of collaborations that can be fun between clinicians and scientists and bioethics and social science scholars. I work with Laura Esserman and Laura Vantveer on their Wisdom clinical trial, which is a large trial to try and use genomic predictors to target the correct frequency of mammography in a large population of women in California. That's a PCORI funded study, which is part of the Affordable Care Act funding. We got an additional NCI grant to be embedded with that group, because this is the first project that is really offering information about highly penetrant genes associated with breast cancer to an unselected group of healthy women. Okay, she's giving you 1 minute. That's good. I'm on the last point. And then finally, the other project that's going on right now, which is fun, is through something called the CSER Network, Clinical Sequencing Exploratory Clinical Sequencing Evidence Generating Research Network. And this is the only sort of data slide that I have. This is just an example of what our CSER project looks like. It's a large multidisciplinary project. We have a pediatrician, there are 4 PIs, a sequencer, a pediatrician, an OBGYN, me as an ethics person, and we're actually offering genomic sequencing to in the prenatal context, and with ill children who have a particular phenotype that we suspect is genetic, and it targets a diverse population, because we all know that patients from diverse backgrounds have not gotten into our genomic databases, so there's a lot we need to learn about how to do this. And then my team, and the line across the bottom here, that's the right one. We're actually doing in-depth interviews with a subset of the participants in this study right after they get the results of the sequencing, and then a follow-up at 6 months, just to get more of a sense of the impact of this, so that we can understand how it affects different populations. So this is my last slide. There are a couple other future collaborations that I've been thinking about. We've been trying to develop some relationships with nephrology because there's some really interesting issues that we're talking about. There's one particular genomic variant called APOL1 that is associated with kidney disease, which is found more frequently in individuals of African ancestry. So that's very interesting in terms of how we would use that information. And the Apollo network that was just funded here at UCSF is trying to think about how you would think about that variant in making decisions about transplantation, either for donors or recipients. And then finally, the one other thing that I would love to do more of is work on big data issues, and in particular the governance of all of the big collection of UC Health data, which we will all have some access to. Lots of issues come up. When should we share that with for-profit entities like Google? How should those kinds of transfers of data and information be overseen and governed, and what should be the role of the patients? You know, all of us will have our data in there too. Do we want to have a role in that? So that's so thank you very much for the opportunity of speaking.
Welcome back. We next have Dr. Tung Nguyen.
Good afternoon. My name is Tung Nguyen. I'm a professor in the Department of Medicine. Research-wise, I'm the program leader of the Cancer Control Research Program at the Cancer Center. And I'm mostly here as a Dean's Diversity Leader in the School of Medicine Differences Matter Initiative, and I'll tell you what that is in a second. But my talk is about why we should think about diversity and disparities in our work. And you know, generalizability is a big word that we use in our study reviews, for example. So this is the distribution of the United States population, 2015. If you have a cohort or a study that is mostly non-Hispanic whites, your result's only applicable to about 60% of the population. And if you have a brilliant idea now that will come into fruition in 10, 20 years, and you work with only non-Hispanic whites, you will be conducting minority health research, because there are only 43% of them by 2060. So that's why I think from a scientific point of view, I think we should care about this. Another thing that's really important is that quality and outcomes. We are working in a health research center, not just pure basic science. We care about things like health outcomes. In my field, cancer, here's the graph of 5-year survival from cancer, people who have diagnosed with cancer. Red is white, blue is black, for the last 40 years. And what you're seeing here is really the progress of what a lot of things have happened: smoking cessation, tobacco control, basic science research, clinical care. Everybody's doing better. However, if you look at the gap, it hasn't gotten much closer. And so my point is that you don't have quality without equality. I can give you an example of many systems through which one group is doing really well, one group's doing badly, and you have an average of great quality. And we wouldn't look at that and say that's a quality outcome. So without thinking about disparities and diversity, I think we're not going to get to where we want to be, which is a healthcare system that actually takes good care of everybody. So why this is important to a lot of what you do, most of which I didn't understand, but here I am. I'm very envious of you guys. Have such great terminology. Community. I don't work in immunity. I work on community. And I'm very jealous of proteomes and genomes and all these things that you guys have. But here are some of my ohms. I work on income. I work on home. I work on overcome. And I work on outcome. And the question here is, are we interested in precision medicine? Or are we interested in precision health? Because these are the most important determinants of health, scientifically speaking, okay, and health outcomes. So for me, I was told to say a little bit about what I do. Here's what I do. This is what it feels like when I do research. You guys have seen the Shawshank Redemption when he tries to dig out the wall with a spoon? It's like, well, that's me doing research, looking through a little hole, trying to figure out what's on the other side. Did that for a while, and I figured out what I don't want to do is keep on doing that. So I actually now do mostly trying to figure ways of going around the wall, getting a bunch of people to climb over the wall, or finding a few hammers and knock down the wall. with a bunch of other people. And the reason why I do that, this is my mom from many years ago. The picture is her standing in front, in the middle of the snow in Pennsylvania. And what you don't see here is that a couple things. One is that 6 months before this picture was taken, she lived in a tropical country, doesn't speak a word of English, and didn't know what snow was. The other thing that you might notice is that she's wearing a mini skirt underneath that big winter overcoat. And my goal in life as a researcher is to enable people to be able to wear mini skirt under winter overcoat in the snow. That's what I do. So for example, this is a project we just completed called Health Within Reach, using mobile technology to improve Asian American health. Big thing is just the background of health. Quickly, hepatitis B, big problem with Asian Americans. Most of you probably already know that. We did this project where we built this app. As you can tell, I have lots of people working with us, doing what we call a technique called stakeholder input, which allows us to think about how to actually influence the people we're trying to influence. We designed an app that's pretty easy, clean, simple to use, multiple languages. And then we actually end up recruiting 450 Asian American patients here at UCSF in the general. And a lot of people are like, well, you know, we can't recruit minorities. We can. And then we did a randomized controlled trial with them that showed that the app worked. So what is the relevance of this to you? Well, the School of Medicine, under the leadership of Dean King, has really made diversity and disparities a fourth pillar, right? So we have research, education, and clinical care. And now diversity, equity, and inclusion is the fourth pillar. And we created the Differences Matter Initiative. There are 6 different working groups. I lead, along with Carol Gross in the graduate school, the research aspects of it, which means everything that has to do with research. And we call our group the Research Action Group for Equity, which is short as RAGE. So what have we done and what can we do to help you? We've done a little bit of work with the CTSI on clinicaltrials.ucsf.edu. So if you do any clinical trials, your studies will show up here, and we try to turn it into easy for people to use and in multiple languages. We actually have done a symposium on minority accrual to research, and hopefully we can have more of those in case you're interested. We have a CTSI consultation service on minority accrual, so if you are working with human beings, then please, and you want to get more minorities. It's actually very hard, if you don't work with minorities, to think about working with minorities. Everybody says it's so difficult to recruit minorities. It's not. It's just as difficult for me to go to your lab and do your work. We have lots of people doing that kind of work, so you don't have to learn it. Just find out people who does it really well and collaborate with them. But don't say it's not. Don't say it's difficult, because if you do that, then you're blaming the people. You know, the resources are there. We have some of the best diversity and disparities researcher in the university, in the country, in this university, and you should never have to feel that way. I'm part of the CTSI Community Engagement and Health Policy Consultation, so if you want to go into the community, you know, you may ask, basic science, what does it do? There's always a way to engage stakeholders in your research, and we can help you with that. If you have no interest or no possibility of working with minority patients, we do believe that having more minority researcher is a good thing. I know that basic scientists do apply for a lot of diversity supplement. We want more diversity supplements. So if you have a grant that's eligible, and that's pretty much any NIH grant, please think about applying for a diversity supplement if you need help with it, or if you need people to fill those spots. You can talk to me, and I'll try to connect you. As you know, we do have precision medicine cohorts here. So at UCSF, the leader of that recruitment effort is Bob Hyatt in Epi and Cancer Center. And the LGBTQ network in the country is here at UCSF too. Mitchell Lund in nephrology and Kirsten Bibbins Domingo in general medicine and Epi. So we're here. If you're interested in any kind of work that has to do with diversity or disparities, please give me a call, give me an email. So thank you very much. And I don't have high-tech stuff. I just have low-tech stuff. But I do want to end by saying you can go faster alone but farther together. Thank you.
Thank you. And now please welcome Dr. Sam Pleasure.
Hi, thanks for inviting me to talk today. So I'm going to be talking about a project that we've been working on for several years now. My lab, along with Joe DeRisi's lab especially, and Michael Wilson's lab. Michael was a postdoc in Joe's lab originally and now has his own laboratory in the Department of Neurology. Essentially, we've been working on trying to improve ways to approach autoimmune encephalitis. And the project really started out with Joe's work in infectious encephalitis. And so the three of us, Joe is obviously an expert and world's expert in metagenomic approaches to infectious diseases. Michael and I are both neurologists. I'm a developmental neurobiologist, and Michael's a budding immunologist. So we've sort of come together to work on this project. And so to introduce the problem a little bit, so we're focused currently and for the Marcus Project on autoimmune encephalitis, and this is really a major clinical problem. About 20,000 patients in the U.S. have encephalitis every year. They're responsible for billions of dollars of inpatient high-level care. Many of them are ICU-bound for very long periods of time, and they can be very, very sick. So they're very expensive patients. The diagnosis is very, very challenging for neurology services. About 50-50 of them, about half of them are inflammatory and half of them are infectious. The data for this is actually very, very poor because the ability to detect infectious causes is pretty poor. But this is estimated based on some studies. The time to diagnosis for these problems is extremely critical in their care because and many of the diagnostic tests that we do take 1 to 2 weeks in their current form. So that you're sort of stuck in this difficult situation of you're taking care of a very sick patient and you send off a bunch of tests and then you wait and you're trying to figure out how to treat them. The therapies for autoimmune causes, autoimmune sources and infectious sources are obviously quite different, and they can be very counterproductive to each other. You may be tempted to immunosuppress a patient who you think may have an autoimmune encephalitis, but at the same time be worried that they have a parasitic process or something like that. And so you may be getting the patient into extra trouble or be giving them a lot of empiric therapy when it would be much better if we could diagnose these things more directly and more quickly. So this is a sort of a case flow chart that we've developed through a project, sort of a precision medicine project that includes a research, sort of predominantly research arm, but now there's a clinical arm that's actually run by Charles Chu and Steve Miller for infectious causes of encephalitis that's up and running and is actually now a diagnostic test that can be done at either Parnassus, the VA, or the General, or outside hospitals. And essentially what happens is, and this is the way we conceived of it for our research program, is that we have a patient who joins the cohort, which we call the NID cohort, which stands for neuroinflammatory diseases, and that we sort of have an email triage for whether we think it's likely to be infectious or autoimmune, or we can't tell. And so then it either goes into a pan-pathogen discovery approach using metagenomic sequencing in Joe's lab, or an autoimmune discovery approach, which is also partly in Joe's lab, partly in my lab, and partly in Michael's lab. And then we come back together with a sort of multidisciplinary board, which meets monthly, to discuss interesting cases. And this was modeled in part on cancer boards, on tumor boards, where multidisciplinary people come together to discuss this. And in this case, what we're doing is trying to come to some useful conclusions using these research tests, not yet diagnostic and not yet for clinical purposes, but we're trying to sort of head there. So what do we do? So for the autoimmune discovery part, and obviously I only have 5 minutes here. So one of the things one of the first threshold tests we do is we check whether we believe the patient has autoantibodies in their CSF. So we use their CSF essentially as a staining tool for mouse cortex. And this is an example of 3 different patients from our NID cohort who have very distinct staining patterns when we just use their CSF as a primary antibody and stain tissue. And you can see that it stains very different kinds of cells in different places in the nervous system. We do this very systematically. we also take their patients, if the patients are positive in this screen, we use their CSF to do an IP and mass spec. And we also, in selected cases, but the thing that's really new, which was developed in Joe's lab by a graduate student, is to use a phage display screening approach, where they've generated a phage library that's completely overlapping the entire human proteome of 700,000 phages, essentially, that is 49 amino acids long and with 24 amino acid overlap, and essentially use that to screen for linear epitopes. We're experimenting and looking forward to whole protein approaches or eukaryotic expression approaches that would allow for post-translational modifications. This allows for rapid rapid evaluation. Essentially, you know, as I think of Joe, I think he thinks about sequencing and he tries to reduce everything to sequencing. And it's brilliant. It's a brilliant way to reduce everything to sequencing, I think. And so essentially, you just run a big sequencing reaction, you get the answer. Now, this obviously isn't perfect, and we're working through it. So we, you know, we've now had some positive hits. We're working on some more, and we're working on a paper that has about 5 or 7 positive novel hits. The immediate goals for the next couple of years are to continue screening using tissue phage display and IP mass spec. We're developing an anatomic atlas of staining patterns for all these patients, where we have comprehensive sections through the entire mouse brain that we will be able to share with the community to look at different patients. We're generating recombinant human monoclonal antibodies for the pathogenic antibody targets so we can use them to generate animal models. In my lab, we're using antibodies to known targets that other people have found to look at the pathophysiology in animal models, and we've made a lot of progress with NMDA receptor antibodies, which I can't tell you about today, but that's one of the main syndromes is anti-NMDA receptor antibody syndrome. And then we're working towards the idea of having a sort of a UCSF autoantibody test where we could multiplex screen for all the known autoantigens, because we already know about 30 of these. We think there are a whole bunch more, but a test that could screen for 30 of them in a day or two would be dramatically helpful. Right now, each test is a send-away test that has to be done separately, and it takes a long time. So I'm going to just skip this, and that's it. Thank you.
Thank you so much. Now please welcome Dr. Dean Shepard.
Thank you. So I'm the third speaker from the UCSF Pulmonary Division. And I'm going to talk about a project that Wendell Lim and I have just gotten started working on to try to engineer lymphocytes to treat tissue fibrosis. So basically, our goal is to extend T-cell-based therapies, which have been shown to be so dramatically effective for cancer, to non-neoplastic diseases. And we're starting with tissue fibrosis because tissue fibrosis is poorly treatable, and it's responsible for about 40% of severe organ dysfunction in chronic diseases. And the mortality rate for some of the most severe forms of tissue fibrosis, like pulmonary fibrosis, is actually worse than most cancers. And the general idea that we're using is a tool that Wendell's developed initially for engineering T cells to treat cancer that involves taking advantage of the normal Notch signaling pathway, where Notch recognizes a tethered ligand, and when that tethered ligand is bound, there's a conformational change in the transmembrane and cytoplasmic domain that reveals a cryptic proteolytic site that's cleaved by a gamma secretase, releasing a transcription factor in the normal Notch that then drives Notch transcriptional activity. And in the case of a synthetic Notch receptor, the only thing that's saved of Notch is this transmembrane and proximal cytoplasmic domain. The extracellular domain is replaced by a detector that can detect anything, usually a single chain antibody. And the transcription factor can be replaced by any transcription factor, and that then allows us to transduce the same T cells that express this synthetic Notch receptor with a construct that can drive any gene that you want. So we can basically develop any therapeutic that can be encoded in the genome to be delivered and made only when the T cells are encountering the appropriate target. And so to do this for pulmonary fibrosis, we started with data from single-cell RNA sequencing that my lab's been doing for a few years to characterize the cell populations that are present in multiple models of tissue fibrosis. And this is just showing one example where in pulmonary fibrosis induced by bleomycin in mice, we can see this pattern of lots of different kinds of cells. So these clouds are each hundreds of cells. And you can see here, if we just look at collagen expression as a marker of the cells that might be important for driving fibrosis, we can identify several genes encoding cell surface proteins, for example, cadherin 11 or PDGF receptor alpha, that also mark the same population of cells. And these two populations down here are actually pericytes and smooth muscle cells. So all of the fibroblasts are marked by these cell surface markers. And then the general strategy that we've used is to generate synthetic Notch receptors that have on the extracellular domain single chain antibodies. We're starting with cadherin 11 and PDGF receptor alpha. We're using the yeast transcription factor GAL4 as a test system. So when this receptor gets ligated, GAL4 is cleaved and then drives any genes that are expressed the UAS promoter that's GAL4 responsive. And so we've begun by trying to target the TGF-beta signaling pathway, because TGF-beta is known to be a central driver of tissue fibrosis. But if you inhibit TGF-beta globally, there are many desirable functions of TGF-beta that are inhibited. So this allows us then to localize TGF-beta inhibitors to the surface of these T cells, or to be secreted locally just when those T cells are activated, when they encounter activated fibroblasts. And we have a very simple readout to see whether this system works, which is just phosphorylation of a transcription factor, SMAD3. That's an early step in TGF-beta signaling. So as I said, we've generated as detectors these single-chain antibodies, and we have a series of payloads that we've engineered that we can now express in T cells. So this has been an exciting project because it's allowed us to combine the expertise of the LIM lab in modular design of T-cell therapeutics with expertise in my lab in models, analysis, and treatment of tissue fibrosis. And we've also recruited help from Jim Wells's lab to generate the antibodies that we're using as detectors of the targets we're interested in. And because Jim's lab has a phage display system for generating antibodies, we can get antibodies that recognize both the mouse surface protein and also the human protein, so we can test these systems in mice and then eventually use them in people. And our next steps are pretty obvious. We need to validate the effectiveness of our payloads and detectors in an in vivo system and demonstrate their effectiveness in suppressing fibroblast expansion and pulmonary fibrosis. Initially, we're going to do this in mice. And we want to then extend this strategy to targeting pathologic fibrosis in other organs and also in tumors, actually, because fibroblasts in tumors play an important role in driving the response of tumors to therapies. And we also actually, with our single-cell RNA sequencing, are identifying multiple subpopulations of fibroblasts that play different roles in different models of fibrosis. And so eventually we should be able to design this system to be much more precise, to target just subsets of pathologic fibroblasts. And really this is just a proof of principle, because once we demonstrate efficacy for treating tissue fibrosis, the general idea is that a similar strategy could be applied to a wide variety of other severe chronic diseases which currently have poor therapies, where protein therapeutics might be useful if we could get around the limitations of their normal short circulating half-life and off-target effects at other sites in the body. So that's all I had to say. So thank you very much.
Thank you. Now please welcome Dr. Avanti Shah.
Hi, so I'm giving this talk on behalf of my lab PI, Dr. Sweet Cordero, who is out of the country right now. And this project is done in collaboration with the Weissman lab. So our project relates to osteosarcoma, which is the most common primary bone tumor of children, adolescents, and young adults. We see about 400 cases annually in the US. Localized patients have actually fairly good 5-year overall survival at about 80%, but patients with metastatic disease do much worse, with an overall survival of about 20%. 20% of osteosarcoma patients have metastases at diagnosis. And unfortunately, the localized patients as well as the metastatic patients receive the same therapy, MAP, methotrexate, Adriamycin, and platinum, along with surgical management. And we haven't really moved the needle at all as far as treatment is concerned for the last 3 decades. We've been using these same 3 drugs. And so one of the things that our lab is looking at investigating is how we can find targeted therapies for these patients. And we mainly do this using patient-derived xenografts. So we take a primary patient sample during a surgical procedure and submit that tumor sample for whole genome and RNA sequencing to identify alterations in known cancer genes. This can then inform us about potential targeted therapies. We take the same tumor and put a small piece in an immunocompromised mouse and wait for anywhere from 1 to 12 months for it to grow into a tumor or PDX. We can then take a small piece of that PDX and separate it out for histology and then dissociate the remainder of that PDX. We can then isolate the human tumor cells and store some of it for further studies and then submit some of it for whole genome and RNA sequencing. We can then compare the whole genome sequencing and RNA sequencing data from the PDX to that of our patient's primary tumor. This allows us to ensure that we're actually accurately recapitulating the disease that we see in the patient. We can then use those stored low-passage PDX stocks to generate new PDXs and then treat with those identified targeted therapies to assess the tumor growth inhibition. And so looking at our cohort of osteosarcoma patients, we had 70 different samples for which we were able to perform whole genome sequencing. Each one of these columns represents a patient sample. Each row represents a gene. And then within each gene, you can see identified copy number alterations that are color-coded. So if there are greater than 12 copies, they're represented in red, greater than 8 copies in orange, and greater than 4 in purple. And losses in genes are represented in blue. And so looking across our cohort of patients, we were able to identify these 6 baskets or subclasses of osteosarcoma based on copy number alterations. And they have matched targeted therapies, some of which are FDA approved and some of which are still under clinical investigation. Then we can use our PDX model to model the disease and assess inhibition with treatment. So this is just an example of a CDK4 amplified tumor in a PDX, or in a PDX, CDK4 amplified PDX, treated with palbociclib, which is a CDK4/6 inhibitor. And you can see that compared to vehicle, the treated mice had significant tumor growth inhibition, although the tumors did not completely shrink away. And so across our entire cohort of PDXs, across these 6 different baskets that we've identified, you can see that our tumor growth inhibition is at or greater than 60%, which in some studies has been shown to be kind of the marker that this could have clinical efficacy. So although we had tumor growth inhibition, as I said, we didn't have any tumor shrink away. And the reason we think this happened was because of tumoral heterogeneity. And so we're now utilizing single-cell RNA-seq to assess that heterogeneity. So here we can see this CDK4 amplified PDX, which I showed in the last slide, and you can see that there are 8 different clusters numbered from 0 through 7 based on their gene expression. And you can see that although CDK4 is expressed in all of the tumor cells, it's differentially upregulated in cluster 2 compared to the other clusters. So although all of the cells have CDK4 expression, it might be that in some of these clusters there are other oncogenic drivers that are the cause of disease and the reason that our tumors did not completely shrink away. So how can we find rational targeted combination therapies? Our goal is to use an unbiased approach using Perturb-seq. So Perturb-seq is a technique that will allow us to identify the consequences of gene knockdown by CRISPR-I in multiple clusters for potential targeted genes. First, we generate a pool of CRISPR-I against the most CRISPR-I for perturbations against the most common copy number alterations observed in the PDX. The pool will be infected in the PDX, and then the tumors will be grown in mice with or without 3 days of treatment. At the end, we'll excise the tumors and then form single-cell suspensions and use the 10x genomics platform to identify single-cell transcripts with mapped CRISPR-I perturbation. We will then be able to determine the consequences of gene knockdown on specific clusters in the presence or absence of drug treatment. So then we can study the gene expression dynamics at the single-cell level in response to drug and a CRISPR perturbation. So a hypothetical example is shown below. So you can see that in this CDK4 amplified tumor, there are other clusters which perhaps contain oncogenic drivers like VEGF and MYC. And by looking at the changes with CRISPR-I and palbociclib treatment, we might identify new drivers and identify potential combination therapies of interest. So these experiments are currently ongoing, so this is just a hypothetical example, but we just wanted to thank the Marcus Seed Program for their funding for these experiments.
Thank you. And we have our last speaker, Dr. Claire Brindis.
So I know that I'm standing between you and the networking. It's really a pleasure to be here. I want to thank Keith for his leadership, as well as Gretchen and Paige for this afternoon. And really, it's been a fascinating afternoon in terms of really thinking about how many of you are working on topics related to how disease happens, why does it happen, and eventually finding out why does it happen in inequal or with inequality, as Tung would point out to you. The areas that I've been working on at the Institute for Health Policy Studies really is the last mile. It's that when you've solved these tremendous health issues and eventually it gets into clinical care, who will pay for it? Who will have access to it? Who will be the funder for it? Will government pay for therapy? Will we find that there's different ways of delivering health care? How will your data intersect with the electronic healthcare record in terms of really trying to figure out what are the patterns of healthcare delivery, healthcare response, and treatment for the issues that you encounter? So I feel proud to be part of UCSF, where this kind of research occurs, and really looking for these bridges that occur, and also taking into account the issues that Barbara talked about, which has to do with medical ethics. Oops, sorry. So some of the opportunities that I see with having a policy lens is that you really are focused on a continuum. There's the continuum from bench research, and here some of the policy concerns were really about regulatory issues in the field of precision medicine. Then there's issues of going from bedside to, from bench to bedside, where we really want to look at measuring health services outcomes and payment reform, which is really important. For example, the work that Adam Studley, another pulmonologist working here, who was very concerned about cost, care, value, and ways to reduce the unnecessary use of tests in our country. And then from the bedside, we really think about community and where and how do we reach different communities. There's so many different communities, but really evidence-based interventions, and how do we engage the community voice in the kinds of interventions that's created. And finally, in the continuum, you get to policy, where you inform evidence-based practices for making changes at the local, state, international, and national level. And when I talk about policy, I talk about capital P policy, which has to do with the laws that are passed, the kind of funding, how does Medicare function, how does Medicaid function, how does treatment get delivered. And then there's small p kinds of issues, which really get to be how you translate legislative policy or things like regulatory into your day-to-day practice. Now, some of the barriers that I and you also have encountered is that right now there's really a question about, will precision medicine really deliver? Have you encountered the issue of over-promising and perhaps under-delivering? And I think that this is an issue for a campus like our own, where so many of the resources are being allocated to these very exciting, cutting-edge kinds of opportunities, but also to be thinking about how do we communicate to the public about the importance of your work in doing this very basic research and basic science. We know also that in the public's perception, there are some concerns about the trustworthiness of the kind of research that takes place. And we need to be thinking about, again, how do we shape the message? How do we inform the public about the value and the importance of big data and the importance of pharma in this space, as well as other delivery issues. And right now we know that we're going through a period where hardly anyone likes the government, but it's very important. So we do see that people still love and trust their own doctors, and how do we build on that foundation of trust in ways of bridging what you're doing with the implications for care delivery. So just to finish up, I think that there's so many different opportunities to really understand not only what you do, but the kind of work that some of the social scientists in this room are doing, because we see that there's value added. There's value added when you bring in questions of community. There's issues of integrity as well as ethics, but there's also the policy context. And so I'll give you an example that when our campus applied for the first CTSI grant, I read the RFA and I said to Mike McCune, you know, this is a great RFA. It's going to change the way that we deliver and do science on the UCSF campus, but there's no policy mention. So you're developing a great drug, or you're developing a great approach to overcoming a number of barriers in conducting research. But if you don't have a policy lens, you're not going to really be able to solve some of this in terms of scalability. You might have a lot of barriers. And so UCSF, there were 8 requirements from NIH. UCSF went in with 11 components. The 8 that were required, and then a component for global health, a component for health policy, and then our virtual community. And we got the second highest score in the country because we were uniquely different in terms of being able to bring everyone from bench to policy. So I want to give a shout out for the importance of added value to the research that you're doing. And so we have to be thinking about the ramifications, and I think this is underlined some of Tung's message to you, is how do we assure equal access to groundbreaking science across all populations? Will it be covered as part of health insurance? How will it be reimbursed? And how do we meaningfully engage patients, policymakers, social media, and other stakeholders in conducting precision medicine research? And I have to say that Keith has been particularly instrumental in all of your work, because he has been such an advocate at NIH and in a variety of government offices to advocate for the funding for the basic research that you all conduct. So it is a full circle here. Thank you so much.